Okay, so we're back now for the second half of today's talk. Hopefully it's a little easier to digest in this way if you're watching the video. So we just finished talking about half-lives of proteins, how these are regulated. These are the end products of the central dogma, okay? And then they're involved not only in the end products, but they're also involved in the signaling step in the middle. But ultimately, their purpose is to feed back. I shouldn't say purpose is to feedback. One of their important jobs is to feed back to modify gene expression, because this is a continuing cycle of information going back and forth between genes and proteins in the environment, facilitating life. So I want to spend some time talking about how proteins now communicate to the genes within the nucleus and how they interact with DNA specifically. I want to just make one more point before getting into that. Is on and off throughout the semester, I know I've mentioned housekeeping genes, and we talked about them with regard to as controls for qPCR, and I just want to, since we've also talked about them with respect to these different transcripts, polymerase machineries, I should say, I just want to kind of make sure everybody has a clear idea. Housekeeping genes are cells required for basic cellular metabolism. They're on all the time in all cells, all tissues, pretty much the same levels. There'll be some that may vary slightly from cell type to cell type, but they're generally on at a pretty steady state level. It turns out, this reference is up above is a review article that talks about housekeeping genes, and there's over 3,000 housekeeping genes in humans, and similar number in mice. Some of them are so exceptionally stable in their levels of expression that those are the ideal ones to use for the control. So some of them may vary slightly, but some of them are super duper stable in terms of their level of expression in different cells. And so the importance of knowing what those are is that when we compare gene expression from cell type to cell type, we want a basis. So we'll look at our housekeeping gene that's on at a certain level, no matter what cell it is, right? And then we can look at our gene of interest and see if that gene expression is up relative to the housekeeping gene or down relative to the housekeeping gene. And most importantly, realize that these genes are not the genes that are under type of regulation events that we're about to discuss. So this slide just shows a couple examples of housekeeping genes, and hopefully you're not surprised to see the products of polymerases 1 and 3 here. So you got some ribosomal subunits are pretty prominent. There's one, there's another. So that's not at all surprising. We see some structural protein. This is actually a Pol2 transcript, but it's very stable. It's a housekeeping gene. It's actin, beta-actin, which is just involved in cellular cytoskeleton. And as with beta microglobulin, down here is succinate dehydrogenase, which you might recognize from the Krebs cycle and is involved in electron transport chain in mitochondria. And also down here we got our TATA binding protein, so that's obviously an important gene that needs to be on in all cells at pretty much the same level. And so these are just examples, a few examples out of the 3,000 housekeeping genes. And just again, the point is these genes here are not subject to the types of tissue-specific regulation we're about to get into. So what we're going to do now is start with kind of general ideas and get increasingly specific. So for starters, I want to talk about, again, as a kind of a reference point, is the prokaryotic system. So in prokaryotic systems, the ground state, you could say, is that most genes are on. Polymerase finds promoters. It's pretty indiscriminate. We looked at a few examples, and I said they're kind of tastes of what we see in eukaryotic system, but polymerase is pretty much on. And some of the specificity is modulated by the sigma factors we talked about, but there's not too many of them. But then if there's a gene that you want to keep off and only use at certain times, you actually turn it off. You repress it. And so the lac operon is literally textbook example of that. So we don't have lactose in our environment most of the time. We only want the lac operon to be active when the lactose becomes present. So we have the gene kind of poised to express, but there's a repressor bound there, and we talked about that in some detail the other day. And this is a pretty standard regulatory mechanism in E. coli. The flip side is if we look at eukaryotes, we kind of, the ground state you could say is off. So we have many, many, many more genes, much larger genomes, and they need to be condensed and packaged to fit into our nucleuses. So we do that by chromatin and nucleosomes. So we've talked about that in the past. And the result of that packaging and condensation is most regulatory elements are masked. Okay, genes can't activate them. I mean, transcription factors and polymerases are the genes are unavailable to the transcription machinery. As a result, so our ground state, our default state, is off for most genes, with the notable exception of certain housekeeping genes. And then we have our chromatin remodeling events that can occur to allow access to the regulatory elements that allow genes to get turned on. And we have talked about some about the general transcription factors, and now we're going to talk more and more about enhancers, because these are the guys that are really involved in creating tissue specificity. So this slide shows a schematic from the other day. I hope you recognize. I want to kind of compare and contrast promoters and enhancers for a second. So we've talked a lot about promoters recently, and so what are the defining characteristics of promoters? Is that they can be kind of general, or they can also be tissue specific. They're orientation dependent. So within a gene, there's a template strand, and that template strand is the one that needs to be transcribed. So that means this transcription complex has to be assembled in the right location so that it has access to the template strand, right? So, and then the polymerase is going to proceed in a certain direction. So they're specific. Certain elements within the DNA. They are orientation dependent, and they work at short range. They need to be pretty close. There's components of them that may stretch up a couple hundred base pairs. Some of the TATA-box and stuff is, just maybe 30 base pairs or so upstream of the transcription start site. So that's promoters for you. Short range, orientation dependent, and they're specific though. And there's some modifications to promoters that will make certain tissue specific changes. The ones we're going to start talking about now, and really is where a lot of cool action is with enhancers. So enhancers are tissue specific. So these are the things that want to turn genes on and off in different tissues as cells develop, as cells and tissues respond to the environment. These are orientation independent. So that means, if we take a look at this diagram up here, we have our TF2D binding to the TATA-box here in this promoter. And you can see how nice and close this promoter is to the transcription initiation site. This region up here represents an enhancer element, or an activation binding domain element. And it can be pretty far away. It can be a kilobase away, it can be 10 kilobases away, even occasionally there's some that are 100 kilobases away. So their function is tissue specific, so that means that when you're turning genes on via enhancers, they're done in fashions determined for the development of that tissue. And also this element can either be in this orientation or this orientation relative to the promoter, because they're not palindromic, they're not such simple things, and still be perfectly effective in activating this promoter to be more transcriptionally active. So those are the defining features of enhancers: tissue specific, orientation independent, and act at a long distance. So now what I'm going to do is kind of go through the sort of theoretical ways that different promoters and enhancers can interact, and realize that there are pretty much examples for each and every one of these. But I just kind of want to, this is based on the image diagrams in your textbook, so I just want to kind of stick with that, so it's, you can read through it. The terminology here they're using is a regulatory binding site. I might call that an enhancer. They're kind of interchangeable, and you'll see this slight difference in a little bit. The protein that binds to a regulatory binding site they're listing here as a transcription activator. I may, in my discussion, refer to it as a transcription factor. Same difference. Here's your dashes for space. Here's your promoter in purple, adjacent to the transcript. So we'd have our promoter, orientation dependent, proximal to the promoter, to the transcription initiation site. And so the first and simplest way that an enhancer can interact with a promoter is pretty direct. Basically, you have your binding site, the transcription factor binds to it, and that binding site then manages, it's either large enough or relatively close enough that it interacts with the initiation complex. And remember, the transcription factor, while it's binding to DNA here, the other end, it has to be interacting with protein. It's not interacting with DNA here generally. It's stimulating the polymerase to go and be more active. So you can simply have a direct interaction between the regulatory site and the pre-initiation complex. Modification of this, and it would be seen in the cases where some enhancers can act at much larger distances than others, as there's actually a little help that's provided by some factors that will be regulating the structure of larger orders of DNA, of chromosomal loops. So this is an example of maybe it's 50 or 100 kilobases away from the gene it needs to regulate, and there are some examples of that, in which case we talked a little bit about the way DNA in higher order chromatin structures can form these large loops. And so if the loop is structured in such a way that it brings the transcription activator close to a promoter, then you can have interaction between the transcription factor / enhancer and the RNA polymerase by initiation complex, elongation complex. So they wind up having a direct interaction, but it's mediated by larger structural changes in the DNA. Another common type of interaction we see is that we'll have RNA being driven off a promoter, an RNA polymerase being made here, and then there's an activator some distance away. The activator's interaction with the RNA polymerase is mediated by another transcription factor or cofactor. So the cofactor may or may not bind DNA itself, but it will mediate an interaction between the activator and the RNA polymerase initiation factor. So you can see here one possible outcome is that as you simply go from off to on. So let's say this polymerase wouldn't function in the absence of this activator, so it would be off. A more likely scenario, or equally likely, is that the polymerase this promoter may be active and be active at a low level. And you have your activator protein bound elsewhere, 5 kb away or so. And upon stimulation of the co-activator, so that is now present and able to create a bridge between these two, we've actually gone from low to high. So we actually aren't necessarily going from off to on as we may be going from low to high. So here we're going from like one strand to high level production here. So that is, either of those are reasonable outcomes through that situation. The converse of that is having an active promoter with a polymerase on it, and maybe it's got either a higher basal state or a gene that you just need to be ready to turn down. And so instead of allowing the activator to act, the cofactor that's going to be involved in this regulation will come and interfere with the interaction between the activator and the polymerase. It blocks the interaction. And so instead of going from off to on, it does the opposite, and it removes the stimulating signal from the polymerase. So you go from on to off, or again, an equally likely, if not more likely, scenarios that you go from high to low. Okay. So I want to look a little bit, move a big picture second here, and look at the juxtaposition of these elements again. The coding sequence, and we went through the definition already, and as a reminder, so here's our coding sequence here. Here's a promoter, so we can see it short term, and remember it's actually going to be orientation dependent too, so that's got to be running in that direction only. The activator binding sites can be at either end of the gene. They can be in either orientation, so this could go yellow, pink, blue in there, or it could go blue. It could run in the orientation, and we can put the blue out here, and then have the pink. then yellow, and would this be just as active? Okay, so it's orientation independent, and again, there can be shared binding sites at both ends of the gene or distinct binding sites at either end of the gene. But it's important to realize, and what we're going to start talking about now is that there is these DNA protein interactions are in fact quite specific. There's consensus sequences that have been determined for these things. They're not absolutely perfect like restriction enzyme sites. We've seen this idea that there's consensus elements in DNA and there's room for little tweaks and variation, wiggle room. But they are specific interactions between protein and DNA. Okay, and once again, just to emphasize the point, we can see how important the combinatorial use of these elements is in determining gene expression. So on the top here, we have kind of a theoretical models of some genes, and they're indicating that this is from your book, so you can see there's indicating that there's 5 different A, B, C, D, E, F, and G are different DNA binding proteins, and they have these different target sites that they can interact with, and depending on which target site is in front of which gene, you change the specificity. So we can repeat B, C, and E work to activate gene Y in combination with F and G, whereas A, B, C, D, and E work together to activate gene X. And this idea, this is again another example, this is in a theoretical format of that combinatorial use of information via DNA elements and the proteins that interact with them. And we saw a great example of this when we were looking at really in the promoters already. So now we actually know that TBP has to bind in each and every kind of promoter, right? We saw that. And part of TBP, the specificity, tandem binding protein binds to a tatabox, like shown here. But in the context of these sequence elements, you get 5S RNA transcription. In the sequence of these transcriptional elements, you get tRNA transcription. In the sequence of the ribosomal transcript and SL1, in the SL1 construct, you get ribosomal transcription. And in the context of Pol2 genes, you get various Pol2 gene transcription. So TBP can work and interact with many, many proteins in different combinations to create different types of gene expression patterns. Okay, this slide, which I skipped in the live version, I'll touch on for a second. It's just another way to try to visualize the combinatorial use of transcription factors. And I don't know if it'll help you or not. The point here is that all of the little colored circles here are all different transcription factors. Some of them, we really haven't talked about any of these yet, but they are all our specific transcription factors. Anything with the name oct interacts with a known sequence. It's a consensus sequence called octamer. It's 8 base pairs. It's involved in the regulation of lots and lots of genes. And so what you can see, among other things, is that octamer has several proteins that are distinct from each other that will interact with this 8 nucleotide sequence. And then also, they'll interact and here's OCT1 binding to the octamer. So this is, I don't remember the sequence off the top of my head, but it's 8 nucleotides, and here's the octamer. What octamer is bound, it can interact with octamer bound on other genes. It can interact with OCT6 bound on other genes. It can interact with OCT4 bound on other genes. So you may have multiple octamers, and they'll interact with different octamer protein variants in different combinations. Octamer can also interact with the SOX2 transcription factor. You can flip that on its head and have SOX2 bind to the DNA, and think of it from the end of the SOX2. And SOX2 has got the capacity to interact with OCT1, OCT4, and PAX6. So that, like, now if you had a gene that had an OCT1 site in it, and a SOX2 site nearby, you might see one pattern of gene expression. If you had the same thing that also had OCT1 plus SOX2 plus PAX6, you get a different pattern of gene expression. And that's just kind of a slightly more specific look at combinatorial use of regulatory elements. Okay, but now I want to talk more about the actual interactions of the protein to the DNA itself. And so that's what this slide's about. So when we think about primers and sequences and polymerase, we're thinking about the nature of specificity that's being determined at the interface of the hydrogen bonds within the inside of the double helix. But that's not what transcription factors see in DNA binding proteins in general. They see the whole complete double helix. And you remember we spent some time talking about the different forms of the double helix. Okay, there's our B-form double helix, which is the most common one physiologically. And it's characterized by having both a minor groove and a major groove. So let's say this is our major groove right here, and this is a minor groove next to it. So it's going to alternate major and minor groove. And depending on where the nucleotides, which nucleotides are where, they're showing different side-on edges to the DNA. So, and the shape of what they show, it depends on whether it's in a major groove or a minor groove, because they're not exactly straight. They're kind of pitched a little bit. And so there, you can see that there's a specificity that's determined by each nucleotide pair and its location in the DNA structure itself. So you can see with AT, for instance, if it's in a major groove, you're going to the proteins that interact with it are going to interact with this methyl here and this hydrogen sticking out here. If it's in a minor groove, it's going to see an oxygen and another hydrogen here. But if it was a TA, it's going to be something a little different. A TA in a major groove is going to look like this. In the minor groove, it's going to look like this. So the location of that oxygen is important. That's a big charged molecule. So the main point is to remember that the transcription factors are not interacting with the nucleotides based on the face they interact with each other in terms of base pairing, but there's distinct structures associated with the intact DNA molecule and what is sticking out on the edges of the molecule. So now I want to talk about specific classes of DNA binding proteins. There's several of them, and I'll just do what we did today. So we're kind of a little bit in sync, I guess, that way. And kind of how they interact, and there'll be some examples in here to hopefully give you some grounded examples of things. So the first one I want to talk about is called the helix-turn helix. It's not a very exciting sounding name, and it's based deliberately on its structure. So here's the protein. There's helix 1 shown in green. This kind of slightly disordered region here in yellow that allows a little bend in the protein, and then another helix. And you can see this red helix. You got to think of it as coming in and out of the plane of the slide. And it's engaged in the major groove of the DNA. And that red helix is the DNA binding domain. And a lot of bacterial DNA binding proteins use a helix-turn helix motif to create their specific regions. This supposedly is the lac repressor. I think it could be pretty much anything. There's nothing too diagnostic on it. But 2 helices, one of them is a DNA binding domain, and it kind of fits into the major groove of the DNA. The next one I want to talk about is the homeodomain. So this is a protein domain that comes from studying development, and as organisms grow, they kind of add segments. I think it's known with certain insects. It's very predominant, but it happens we have these proteins derived from the same type of class of orthologs, and the homeodomains, instead of having 2 helices, they have 3, which is indicated here. So you can think of it as helix, turn helix, plus 1. And you can see 3 helices up here in this diagram. You got your red helix that's still maintaining its kind of being stuck there in the major groove, and you can see it here on this side of it. You got a black helix and a green helix. So the final thing that the homeodomain proteins include, though, is an N-terminal tail. And you can see it in this diagram on the top and the bottom, coming off the black. Which color will show up the best? It is kind of reaching across the minor groove and kind of hugging the DNA. You can see that also. I've just put a couple other versions of the same diagram up there to help you visualize this a little more easily. So in this model down here, you can see your 3 helices again, and then you can see the N-terminal tail reaching across the minor groove right there. This particular protein model down here is this is a homeodomain plus a transactivation domain. So realize that when we're talking about the homeodomain, we are talking only about the helix turn helix plus the tail. That's the homeodomain. But a homeodomain will be found in proteins with other functional domains. It may be a transactivation domain, it may be a kinase domain, any type of domain that homeodomain needs to work with to make that particular protein able to achieve its job. So the homeodomain is just the part here interacting with the DNA. And this is one more look at it before we move on. I found this picture actually, I think, maybe the clearest of all to see. Again, you got your a major groove here. You got your 3 helices. You can see the DNA binding barrel in the major groove, and you can see the N-terminal tail reaching across the minor groove. And this shows a couple of the charged amino acids and their interactions. Okay, the next one I want to talk about is the leucine zipper, another very popular DNA binding type of protein. And it's called a leucine zipper because it has a leucine zipper in it. So leucine zippers are each by definition, they have to interact as dimers. They're either homodimers or heterodimers, and we're going to see that in a second. And those dimers are held together by virtue of this region. One of the tails is the zipper region, which you can see here. It shows the red. And the zipper region is characterized by these evenly spaced leucines. And what that results in is that they're always showing up on the same side of the alpha helix. And then the other part of the complete protein includes the DNA binding domain. So the zippers hold the 2 proteins together, and the DNA binding domains then go in and kind of act almost like chopsticks or scissors to hold on to the DNA. This leucine zipper is based on the fact that it's an amphipathic helix. I don't know if you guys remember that, but that means that there's different charged hydrophobic and hydrophilic molecules on different sides of the helix. So if you have all the lysines on one side, that's your hydrophobic side. And then the other side of the helix will be your hydrophilic side. That's going to be exposed to more things in the aqueous solvent around the DNA. And since you have 2 helices interacting and they both have this hydrophobic face, that's what encourages them to interact together. That's the 2 hydrophobic faces of the leucine zippers drive that's the energetically favorable thing, is for them to be zippered or velcroed together. Okay, so here are examples of leucine zippers in action. So the first one I want to talk about is CREB. That's shown here on the left. This stands for cyclic AMP response element binding protein. Cyclic AMP, I think I misstated this during live lecture. Cyclic AMP is an intracellular messenger. So in the vein of the JAK-STAT very simple signaling pathway I showed you, cyclic AMP is involved in many, many intracellular signaling pathways. And there's DNA elements in protein that respond to various gene activation cascades, and this is the protein that is there's like kind of a correlation between cyclic AMP signaling and certain genes being on and certain genes having. I mean, once the motif is defined by one particular experimental system, the name's stuck. So It's called the cyclic AMP response element, and it's pretty well distributed throughout our genomes, and this is the protein that can bind to it. And so it is a leucine zipper. And so I think this makes a really clear case of how the leucines interact with each other. So they're on that internal face of that amphipathic helix. And what they drew here is I think they actually drew out the carbons of each leucine. So because, it's a 4-carbon R group. And so you can see how they really interact together. And this is that hydrophobic plane holding the leucine zipper of the leucine zipper domain together. And then the other domains on the protein are the DNA binding domains, and you can see them down here kind of holding the DNA like chopsticks or tweezers or whatever. Now, leucine zippers can also hold proteins together that are not the same protein, so that would make them a heterodimer. So that example is shown here. Heterodimer. And the example I have of that is this transcription factor called AP1. And AP1 is composed of 2 different distinct proteins, Fos and Jun, but they, similarly to Krebs, interact via a leucine zipper. I'll share with you a really cool experiment that then kind of brings us back to GFP and variants and why variants are made and how they've been used experimentally. So in this case, the first part I want you to look at is Jun and Fos, which are right here, okay? So what they did is they took Jun and they added a piece of the GFP protein on there. So you can see it's not an intact GFP barrel, just most of one, okay? So this thing by itself will not be fluorescent. Likewise, they took Fos and added a different component of the GFP barrel to it. And similarly, that will not be fluorescent. But if these 2 proteins interact in vivo, which we expect that they do, then we will get an intact fluorescent GFP. And actually, it's a YFP. It's this yellow one here. So if it gets together, it'll be yellow. And you can see that if we look at YFP, if we activate YFP directly with the laser, Fos and Jun are interacting, and we can measure the yellow. Well, it turns out that Fos and Jun, AP1, interacts with other proteins, and they wanted to demonstrate that. So one of the other proteins, transcription factors that it interacts with, is called NFAT1, and they made another fusion protein. In this case, it's NFAT1, and they used the GFP variant cerulean to look at that location of that, and you can see where NFAT1, you can directly activate NFAT1 and measure that signal directly. But what FRET is we actually separate out. FRET is, if you remember, we talked about fluorescence in lab, fluorescence resonance energy transfer. And to do that, what we actually do is we want it, if all these proteins are interacting in the cell as shown, then we can stimulate the cerulean, and that's going to emit a certain wavelength. The wavelength that cerulean emits is actually the wavelength required for activation of Venus, the YFP. And you can see that's what happens here. So we only activate one channel of light, which activates cerulean. Cerulean activates YFP. And then when we measure and look just at the YFP signal, we can see that they've interacted. All 3 of them are interacting together. And so that's just a really cool example of how GFP technology is used to study in vivo cell protein interactions. Okay, so next up is going to be the basic helix, loop helix. It's actually, we'll start there on Friday. So I'm going to stop.